What a pleasure to see so many of you here this evening. Welcome this evening to the Italian Academy at Columbia. I think many of you I'm David Freedberg, and I have the honor and the pleasure of being director of the Academy. But above all, it's a pleasure and an honor to see so many of you and so many distinguished visitors here this evening. I take it, in a way, as testimony to the distinction of Vittorio Gallese and of the discovery with which he is most famously associated, that of mirror neurons, that so many of you are here this evening. In a way, it's even more remarkable because what I find when I give 3 lectures in a short space of time at the same place, the audience tends to taper off. One would like to think that every time one spoke, more people came. I think the only person I really know, other than rock stars and sports stars, and even in those cases the audience tapers off. The only person I know who has managed to get an ever larger audience in this room in a short space of time has been Vittorio Gallese. Vittorio, who, as you know, is Professor of Human Physiology at the University of Parma in Italy, and I'll come to his distinctions and the other aspects of his career in a moment. But I think some of you here will have attended the remarkable conference which we organized in collaboration with Eric Kandel in March of 2006, in which Vittorio gave one of the star turns at the Conference on Art and the New Biology of Mind, held here in this very room. When I say star turns, there was stiff competition that came from a number of famous neuroscientists, some local, Joe Ledoux, some from further afield, Antonio Damasio and Ray from California now, and Ray Dolan from London, and a number of other distinguished people, and of course some wonderful artists, including Marina Abramovic, who was much more active on that occasion than she was in her recent stunning performances at the Museum of Modern Art. But Vittorio's lecture stood out for its combination of scientific precision and passion about the possible applications of the discovery of mirror neurons, first in monkeys and then the discovery of mirror systems in humans as well. So then he came again a year later at the insistent invitation of many of our colleagues and many of our students in April of 2007, where he set out more clearly and more articulately than he had time to at the first event his views on the significance of mirror neurons, the understanding of mirror neurons for the understanding of embodied involvement with works of art. Now, of course, many of you here will think that artistic processes, artistic reactions are largely cognitive, but it has been the great merit of Vittorio above all in his reflections on art, because he has extended his neuroscientific work into a wide range of cognitive and social fields, ranging from intention to the problem of autism, and above all to something that he's been passionately involved with, the philosophy of art and aesthetics. And on that occasion he set out the baseline views of his baseline views on the meeting of science and art. In other words, he anticipated by 2 years the 50th anniversary of the publication of that famous book on the gulf between the arts and the sciences, the famous book by C.P. Snow, The Two Cultures. And now he's back again, and it's wonderful to have such a great group of you here in our beautiful theater this afternoon to listen to him. So most of you know about Vittorio, that's why you're here. But let me remind you that he is, as I've said, Professor of Human Physiology in the University of Parma, with appointments in the departments of neuroscience, psychiatry, and psychology. In fact, he took his MD in 1990 and has always maintained a significant interest in psychiatry as well. But he really became known, I think, above all, for his role in the discovery of mirror neurons, along with Giacomo Rizzolatti and his other colleagues at the University of Parma in the late 80s. The first article appeared in 1992, and then the ones that most of us know were really, the results were more fully published in a pair of remarkable articles in 1996. I think Vittorio, of those of you who do not know about mirror neurons, Vittorio will give a brief account of the significance for the understanding of aesthetic responses this afternoon, but in so doing, he will also give you a sense of the further import of those discoveries of the 90s. They were in monkeys, but of course they extended controversially, as we shall see perhaps in our discussions with our colleague Daniel Salzman of the Department of Neuroscience here, that extended controversially into the field of human behavior as well. So there have been a flood of articles from Vittorio's hand dealing not only with mirror responses to actions, but also mirror responses to touch and to hearing. And I think Vittorio has really made his name as the most articulate scientific exponent of the neural substrate of phenomenological responses to art. Those of you, I know there are many of you who are interested in art in this audience, and you know, for years we have worried about the sense that we have when we go into a gallery of being corporeally involved one way or another with the work of art. We have vague feelings about this, but we've never been able to give these feelings any kind of material substrate or any sense that there is a material basis for this. Many of us present have read the works of Merleau-Ponty, which spend a lot of time discussing how viewers have a sense of the artist's actions, for example, in making a drawing or in walking around a landscape by Cézanne. These have always seemed to me to be impossibly metaphysical, and we now have the vague hope. The skeptics will say there still remains very little hope. The optimists will say there is substantial hope. But we do have some hope of better understanding our sense, viewers' sense of their physical involvement, either with the bodies that we see in images, with even the abstract figures that are there, or even more with the actions that produce the marks on a picture. Vittorio and I, over the years, have had many discussions about the relationship between neuroscience and art, and I have to say that in this evening's talk, which is entitled The Body and Aesthetic Experience, a Neuroscientific Perspective, he will refer to a subject about which I have some doubts, at least about the nomenclature of which I have some doubts. In other words, neuroaesthetics. He will talk about the relevance of neuroscience for neuroaesthetics. I'm going to leave it to you to decide how convincing and how useful a term this is to apply to the understanding, the hope of understanding the works of art and responses to works of art through images, through neuroscientific discoveries. Vittorio and I have talked a little bit about this afternoon's presentation, and he has a lot to say, and he very generously, in the course of his discussion, decided to omit a very important chunk of his talk, and I'm going to allude to it very briefly, just to save time, and that is, many of you will think to yourselves, the claims which are being made this afternoon were indeed anticipated not just by neuroscientists, but also by some of the most distinguished thinkers about art, above all in the 19th century, less so in the 20th century, other than thinkers like Merleau-Ponty. And these were, for the sake of the art historians here, amongst the giants in the field, above all, A.B. Warburg and Adolf Hildebrandt, both of whom thought very deeply and concentratedly about the biological aspects of response to visual images. Both of them, Hildebrandt left quite articulate views, Warburg's views typically were rather inchoate. Vittorio has taken this out of his talk this afternoon at my request in order to leave more space to present the neuroscientific dimensions of the subject. So let me tell you that we're going to have Daniel Salzman, our distinguished colleague, who works above all on emotional responses, but I will talk about him when it comes to his turn after Vittorio's talk. But Daniel Salzman will respond to Vittorio's talk, so I'm looking forward to a gripping engagement this afternoon, and without further ado, let me welcome Vittorio Gallese back to Columbia this afternoon. Vittorio.
Everybody, thank you, David, for this wonderful introduction. So, I'm back, and a few of you who may have listened to my talk, which I gave 3 years ago already, more than 3 years ago, will perhaps remember that by speculating on some of the hypotheses we put forward in that paper, which appeared the same year in Intrinsic Cognitive Science, David Freedberg and I said I left you with the promise that those hypotheses would have been put under the empirical test. And luckily enough, now I can show you some data that will conclude my presentation. And of course, I will leave it to you to decide to which extent those data provide any, if any, support to the original hypothesis we started with. We are obsessed, literally obsessed with images, and from the very beginning of a reliable historical record, the proclivity of our species in leaving visible traces on the external world has been discussed in many contexts. Here is an image taken from La Cueva de los Manos in the province of Santa Cruz in Argentina, which dates approximately 7,000 years before Christ. While this is an image which I took personally more recently at the kindergarten of my daughter Leah during the summer party, which concluded the year last June. And it is remarkable to see how the hand represents a crucial aspect of our body that we tend constantly to represent in this very analogic way. But also remarkable is that almost from the very beginning, our species felt compelled to leave a durable trace of its own activity, like this picture taken from the cave of Lascaux, where we see some of our ancestors that are involved in a typical activity, most likely in those times, chasing deers with the bow. These are more recent examples. This is from my hometown, from Parma, Antelami again, Caravaggio, of course, Freud, Bacon. All of these images mean something to us, but beyond the meaning we may recover by looking at these images, which most of the time has a very deep and profound idiosyncratic value, we are moved by these images. And indeed, as already many years ago, David Freedberg proposed, these images, artistic images in particular, exert a very strong power on the beholder. So one crucial question is to determine on which basis this power stems from. Where does it come from? This power is so strong that we are ready to make long trips, pay a substantial amount of money, spending time queuing under the sun or the rain to contemplate these artworks, like this tourist captured by the camera of Thomas Truth in the Accademia in Florence or at the Art Institute of Chicago. Here is a quote I took from an article which appeared in the New York Times a couple of days ago, referring about a nice performance of the Tricia Brown ballet company given not too far away from here, at the Whitney. So Rosalind Solkas wrote, "All the people below surely for a moment imagine themselves leaning straight into the void. And as Mr. Petronio, this guy, made his way down, we understood something about the human body in relation to gravity, the way physical prowess can suspend disbelief, and about the strangeness and wonder of the mechanics of ordinary movement, that no amount of historical footage of Mr. Brown's work or Mrs. Brown's work or reading of old reviews could communicate." So the gist of this passage is that the body is self-explicit, and it anticipates or even make more explicit things that we can certainly describe using language. Here is a quote from a very interesting book by a novel writer, but also writer of essays, Siris Ustvedt. This is a book about her relationship to art, Mysteries of the Rectangle, where she writes, "Visual art exists only to be seen. It is the silent encounter between the viewer, I, and the object, it. That it, however, is the material trace of another human consciousness. In art, the meeting between viewer and thing implies intersubjectivity. The intersubjectivity inherent in looking at art means that it is a personal, not impersonal act." I think this is important because immediately draws a parallel, establish a link between the relationship between humans and artworks and the notion of intersubjectivity. So we can say perhaps that aesthetic experience is a mediated form of intersubjectivity. And this particular kind of intersubjectivity can occur also before or without the linguistic mediation, as maintained by one of the founders of modern theater, Meyerhold, who wrote, "Words do not tell everything." So this means that this aesthetic experience is strongly mediated by our bodily experience, which occurs during the same experience. But one may ask, why do we need neuroaesthetics? Whatever it means, I'll try to specify more precisely what I take neuroaesthetics to be. Why neuroaesthetics? According to many, studying the brain and coming up with these beautiful and colorful images is not so different from indulging in front of a Wunderkammer, where mirable objects from all over the world used to be collected by wealthy individuals in Europe many years ago. And indeed, there is a very important risk in dealing with the brain by means of these highly sophisticated but still very imprecise tools like fMRI or magnetoencephalography. The risk is to reinvent phrenology. So the risk is to reify mentalistic notions into something which has a house in the brain. I don't subscribe to this usage of the neuroscientific method, but on the other hand, I believe that these tools, this methodological approach can shed new light on a variety of topics that not long ago were considered to be completely outside the reach of empirical scientific investigation. So why neuroaesthetics? Many scholars in the humanities are convinced that a neuroscientific approach to aesthetics, while unable to add anything substantially new about art and aesthetics, may hinder, if not even destroy, the magic and wonder normally accompanying our appreciation of art. One problem, perhaps, stems from the fact that so far the interest put by neuroscientists into the investigation of art and aesthetics has been almost uniquely if not exclusively confined to vision. And I think it is questionable, in the first place, to identify aesthetic experience as merely a visual enterprise. But even more importantly, I think it is reductive to reduce vision to a mere visual enterprise, namely to reduce vision to the outcome of the processing of a specifically dedicated part of our brain, namely the visual brain, that here you see represented in these beautiful and colorful maps that comes from the lab of Van Essen. And here you have a more structured, functional version of this topographical map. So the where system, the what system, the motion system, and so on. I think this is a reductive account of vision, because observing the world, and even more when the object of our observation is an artwork, is a much more complex enterprise than the mere activation of the visual brain, because it implies a multimodal notion of vision. Observing the world encompasses the activation, as we have learned through several years of investigation, encompasses the activation of somatosensory and emotion-related components within the more general frame of the intrinsic pragmatic nature of every intentional relation we might entertain with the world. So there is a lot more than the activation of the visual brain in vision than it might apparently look. One example of the intrinsic pragmatic nature of perception is represented by mirror neurons. Mirror neurons were originally described by our group in this region, in the ventral premotor cortex of the macaque monkey brain. This is the frontal pole, the occipital pole, the central sulcus, the primary motor cortex. So these neurons were found within the most anterior part of the premotor cortex. Later on, we discovered that neurons endowed with similar properties were sitting in the inferior parietal lobule in areas that turned out to be reciprocally connected with the area F5, so being part of one of the many parallel parieto-premotor network. Since the very first report that, as David said, appeared in 1992, several more studies were done, and each of these studies enabled us or other groups investigating this particular type of neuron to discover or refine new properties. To put it briefly, these are motor neurons that fire both when an action, a motor act like grasping, holding, manipulating an object is both executed or observed when being executed by someone else. So we proposed that this neural mechanism might embody an abstract motor representation of action. Abstract because it turns out that it doesn't so much rely on the kinematic nature of the movement, be it executed or observed, but rather on a more abstract aspect of our pragmatic relation with the world, namely the goal, the purpose that determines the hand to be shaped in a given way. And from the very beginning, we proposed that such a mechanism could underpin a sort of non-meta representational form of action understanding. Here, I would like to briefly share with you some new results that even you, David, you might not be aware of. The first deals specifically with the point I just made, that these neurons are not so much concerned with movement, but with motor goals. Here you have one mirror neuron that fires when the monkey grasps a small object by opposing the index finger to the thumb, but the same neuron would also fire when the monkey grasps the very same object by employing a pair of reverse pliers, the escargot plier that French people use at the restaurant when they eat escargot. The peculiar quality of this tool is that when you grasp the object, your finger, in order to close the plier, must open. So the goal is achieved here with opposite hand movement with respect to when the object is grasped with a bare hand. Nevertheless, you have the same type of response. So no matter if you close your finger or you open your finger to grasp that object, if you do it with a bare hand or using a tool, the neuron will always fire at the moment in which the goal is accomplished. And what is interesting is that the very same neuron will also respond to a grasp executed in front of the monkey by someone else by using the hand or by using the very same type of reverse pliers. A second interesting result of this study, which appeared this year in 2010, is that on average, at the population level, the motor discharge of mirror neurons is significantly higher when the action is executed by the monkey, the red curve, no matter if with the hand or with the pliers, with respect to when the action is observed. So this mechanism of motor resonance is intrinsically capable of disentangling when the monkey is the active agent of the motor act with respect to when he's merely watching, witnessing an analogous motor behavior instantiated by someone else. So the motor resonance in itself is capable of distinguishing when the action is executed or merely observed. Another very interesting paper came out last December, this time from University College London, from the lab of Roger Lemon. Roger Lemon and his colleagues have been investigating the corticospinal motor system for many years. So this paper, I think, substantiates a major step forward in our knowledge of the property of mirror neurons, because these mirror neurons can be recognized also as being part of the pyramidal tract, which means that the axons of these mirror neurons leave the premotor cortex and go all the way down the spinal cord to target other motor neurons in the spinal cord, which in turn innervate the different muscles, and when they activate, they set this muscle into motion. The interesting novel finding of this study is that a substantial proportion of these corticospinal mirror neurons fire when the monkey actively performs the task, but are inhibited when the action is observed, which means that while during action execution, these mirror neurons are facilitating motor execution by sending their output to the spinal motor neurons, when the action is observed, the neural machinery that enables the observer to act is non-facilitated or frankly inhibited. So this could be an important neurophysiological aspect of the mechanism that enables us to simulate the action we are observing without echopraxically repeating it on the spot. An interesting mirroring property was recently discovered also in the domain of attention. At Duke University, the group of Platt, Shepard, and colleagues described a neuron that not only in the lateral intraparietal area, which is a part of the brain with oculomotor properties, this neuron not only fire when the monkey directs the gaze to a given direction, but also when the monkey is looking at another monkey, gazing in the very same direction. So one could say that here we have an instantiation of mirroring of gazes. So this is quite relevant when we feel, I think, quite compelling when we look at the painting and one of the characters of the painting is directly looking at the beholder. And beside that side observation that might be or not related to aesthetics, I think that these neurons are very important because most likely constitute part of the neural machinery that enable monkeys to share attention, which is an important ingredient of any mentalizing recipe, so to speak. Let's quickly move to the mirror mechanism for action in humans. This is a recent meta-analysis which shows which part of our brain, as investigated by brain imaging, activate both when these different acts are executed or observed. So basically we have the posterior parietal cortex, the ventral premotor cortex with the posterior part of Broca's region, and this region within the superior temporal sulcus, which according to all most prominent handbook is still considered to be purely visual, but apparently it is not purely visual because it turns out to be activated also during action execution or imitation, substantially more than when they're similar, but they are not similar. Not for our participants, but not even for their brain. At the end of the experimental session, all participants were asked to score for each stimulus its aesthetic value. We basically asked, how do you like it? And please give a score from -10 to +10. If and how much movement do you perceive in that image, from 0 to 10? Are you familiar with that image? And if you are familiar with that image, how familiar you are? The participants were all undergraduate students of the School of Medicine of the University of Parma. And it turned out that about half of them never heard the name of Lucio Fontana, and they never saw those paintings, while the other half was more familiar with his work. We performed a repeated measure Anova with the following factors: familiarity, of course, 2 levels, familiar and unfamiliar; hemisphere, right and left, because we record simultaneously from the 2 hemispheres; and conditions, 3 levels: baseline, the dark screen; original, the paintings; and controls, the graphic version. Only condition effect was significant, and we did comparisons, and the result is this. Alpha suppression was significantly stronger, not only with respect to baseline, but also with respect to the graphic modification of the paintings, and most importantly, the same result was obtained when we analyzed the data also with the log ratio, subtracting by the alpha suppression obtained watching the Fontana or watching the modified version, the alpha suppression obtained during the brain activity, the alpha level during rest. So alpha suppression is only evoked by the observation of original artworks, but the most important finding of this study is that it is independent of stimulus familiarity. So it doesn't matter if you are familiar with Fontana, or if you heard the name of Fontana for the very first time once you were debriefed at the end of the experimental session. No matter if you are familiar or not with that artwork, the original artwork evokes alpha suppression, the modified version doesn't. And what is interesting is also that aesthetic judgment is higher for original artworks, regardless again of the familiarity. So participants found more beautiful the original Fontana than the graphic version, although they look very similar. These are the graphs. And movement perception, which is quite outstanding. I mean, I didn't expect most of the people could say yes, because they are watching a static cut in a canvas. Nevertheless, they were able to detect some implied motion in that static image. And that movement perception is higher when observing both familiar and unfamiliar original artworks than when observing control stimuli. But of course, movement perception is higher when the original, when the beholder has a familiarity with that artwork because he knows how it was produced. Finally, we tried to do a correlation between alpha suppression and aesthetic judgment. These are very tricky issues. There have been a paper who spoke about voodoo correlation, when one tried to correlate brain activity with behavioral variables. So I don't think this is probably the most important part of the result. The most important part of the result is that the original artwork uniquely induces motor resonance independently for visual familiarity. But if you want to correlate alpha suppression with aesthetic judgment, this is the only variable that seems to correlate, and it correlates only for the original artworks, but it doesn't with the control stimuli. So, to conclude, the marks on the canvas are the visible traces of goal-directed movements, hints, as we have seen, capable of activating the relevant motor areas in the observer's brain. The artwork becomes the mediator of the motor and emotional resonance that arises between the artist and the observer. The sensory-motor component of stimulus processing represents the most direct and automatic level of processing, which allows the beholder to feel the artwork in an embodied manner. What shall I conclude? What can neuroscience tell us on aesthetic experience, no matter if we stick to the notion of neuroaesthetics or not? I think it can help to rediscover the role of the acting body in intersubjectivity, even if in a mediated way. Embodied simulation generates the peculiar quality of the embodied seen as that plays a significant role in aesthetic experience, and I take it to be one of the main ingredients of our appreciation of art at this basic level that I refer to as aesthetic experience. Why is artistic fiction, this is another, to me, excruciating question. Why is artistic fiction often more powerful than real life in evoking our emotional engagement and empathic involvement? Perhaps because in aesthetic experience we can temporarily suspend our grip on the world. Besides being more focused, besides being in relation with the narrative that most of the time doesn't exist when we have sporadic encounter with facts in everyday life, there is also this aspect which I take to be quite important. We liberate new energies and put them into the service of a new ontological dimension, the aesthetic dimension, that paradoxically can be more vivid than prosaic reality. So aesthetic experience of artworks, more than being a suspension of this belief, as we commonly designate it, can be interpreted as a sort of liberated embodied simulation. We look at art from a distance of safety from which our being open to the world gets magnified. So to appreciate art, perhaps, means leaving the world behind in order to more fully grasp it. Thank you for your patience.
Spoken here before last year at our conference on attention. And Daniel Salzman, who is of course a principal investigator in the Center for Neurobiology and Behavior and the Department of Psychiatry here at Columbia, he played a very important, spoke at a very important moment in which he talked about the relationship between the amygdala, that favorite part of the brain that we used to think only related to fear, but in fact, as he has shown, has a great deal to do with the ways in which we learn to assign emotional significance or value to sensory stimuli in the environment. So he talks not only about fearful stimuli, but positive stimuli as well. So he's made a huge contribution to this particular study, to this particular aspect of the study of emotions, the contribution of the emotions to decision making, and in general has refined the techniques of these kinds of analysis of neuronal function and dysfunction in neural circuits as well. Today I don't think he's going to be talking about the dysfunctional aspects of neurons in their relationship to disease, but perhaps he will have some questions to ask about what remains one of the most controversial scientific discoveries of the last 20 years, that of mirror neurons. So Daniel, please take the microphone. We're happy to have you here with us today.
Vittorio, that was a wonderful talk. As always, you push the envelope of your data in such new and exciting directions that it leaves some of us simple-minded scientists a bit in awe. But I wanted to provide some perspective on what I think is a particularly exciting development of research over the last 20 years, 15 or 20 years, which is the delineation and now expansion of the concept of mirror neurons in the brain and what functions they may subserve. But it's a field that I think is still very much in its infancy, and I'll point out that's the case by comparing it to some other aspects of cognitive neuroscience that we've studied. And in part, this is to challenge Vittorio and his colleagues to come up with experiments that might push the study of mirror neurons in that same direction. Now, of course, many, many years ago, when any of us thought about a mental state, or not us, but human beings thought about a mental state and thought about the mental states of others, we really had no access to their mental states. This is like the Wittgensteinian example of a beetle in a box, right? So if you have a particular thought or belief, that's like a beetle in a box that I have no access to. And the discovery by neuroscientists that you could actually insert microelectrodes, these are small little wires, into the brain and snuggle them up into next to individual cells in the brain and listen in as to what those neurons are doing. What makes those neurons fire action potentials? What makes those neurons communicate information to other parts of the brain? This was like being able to pry open the box and look at the beetle to some extent, to be able to start to characterize the properties of individual neurons in the brain and how those neurons are representing different mental state variables. Now, this was extremely exciting. Some of the best examples of this work came in really in and around the 1960s by Hubel and Wiesel, when they began to delineate how the visual scene is represented in the nervous system, how the visual scene that we all perceive is broken down into different components, some components having to do with the form of a visual stimulus, some having to do with the color or the depth of a visual stimulus, others having to do with the motion of a visual stimulus, and the fact that there are specialized neurons to encode these different parameters in the brain, and then stages of processing that take place in order to reconstruct the visual image from these elemental representations. And the really exciting thing about the mirror neuron observation is it says by using the same general technique, by putting an electrode into the brain and snuggling it next to neurons, maybe you're not only gaining insight into the mental state variables of that person, but maybe you're even gaining insight into the mental state variables of a person that person is observing. Right? That's the genius of the mirror neuron hypothesis. It's that we can have a representation of processes going on in our social partners. And initially this was done in the context of the motor system, and as Vittorio has outlined, they've been pushing the envelope further and further to try to understand how this basic concept, the idea that individual neurons in the brain may be representing information about other people's mental processes, has been pushed now to areas like emotion, to areas like empathy, and now even he's pushing it to aspects of what it means to appreciate a piece of artwork as we observe it. And so this is really cutting edge thinking about what mirror neurons may well be doing, and of course then they would have to live in many different parts of the brain, not just in the initially observed parts of the brain, in the ventral premotor cortex or the parietal lobe, but they would live in many parts of the brain depending upon the specific processes that they were mirroring. Now the challenge that I think still sits there for investigators like Vittorio is to actually show that in fact these neurons are used for specific purposes. And you know, he hinted at this, he hinted early in his talk about the steps that have to be taken as you first discover something and then try to translate it into a scientific finding that has legs, that lasts and has a lasting impact. And the first step is really to characterize, the first step when you're studying cognitive processes is to be able to characterize the properties of the cell or system that you're trying to study. And that's really the process that's been going on for the last 15 or 20 years with respect to mirror neurons. And it's the process that I initially described when Hubel and Wiesel and others initially looked at how a visual scene is broken down and analyzed by individual cells in the brain. But I think one of the things that's so powerful about the work that's been done in visual neuroscience is that that work has led to a series of experiments that have demonstrated, at least in some cases, how these neurons are actually used to drive different cognitive and perceptual behaviors. And it's that challenge that I think that remains for scientists working on mirror neurons. And let me tell you briefly the approaches that neuroscientists take in thinking about how to link the properties of individual cells to specific cognitive acts or behaviors. Briefly, what neuroscientists like to see is ways of taking the activity of the neuron and knowing how it's used by the nervous system to produce an act, whether that's a cognitive act or a behavioral act. And there are at least three ways in which you would do that. One way in which you would do that is to simply measure neuronal activity and be able to correlate it with the particular behavior that you're executing. In other words, you want to be able to predict the behavior or the cognitive act that is happening just on the basis of the activity of the neuron. A second major method that's used is one in which you inactivate neurons. You can do that by experimentally giving an animal subject a lesion, or you can do it pharmacologically just by inactivating neurons. And the prediction would be that if you inactivate neurons that are thought to encode a specific property, and the brain is really using that encoding in order to do something, the prediction would be that that function would be impaired by inactivating or lesioning those neurons. And the third major way that neuroscientists would try to demonstrate that neurons that are encoding a particular property actually subserve that function would be to explicitly activate those neurons. This can be done pharmacologically, or with electrical microstimulation, or even with new genetic tools. But the prediction again would be that by manipulating the activity of specified neurons, you can actually cause subjects to have certain cognitive acts or behaviors. And those three types of data actually have been well established in the visual system, and what makes the visual system perhaps the best understood of the cognitive systems in the brain. And in particular, if you think about motion perception or how we see motion, there are neurons in the brain that tell you what direction of motion you're seeing. So if you were to pass a bar of light moving in one direction, a neuron will fire a burst of action potentials. And if you pass the same bar of light moving the other direction, it will not. And there are different neurons that encode different directions of motion. And it's been known from work in animal subjects by me and others, some of my mentors, that if you lesion these neurons, which are called direction selective neurons, you impair motion perception. So that's the first test, right? So we can knock out a particular neuronal type, and then we can assess its cognitive function. It's also known that if you measure the activity of individual neurons that are encoding the direction of motion, it can actually predict whether or not an animal subject on a moment-by-moment basis is going to tell you that motion is in that particular direction. So if you're a neuron that's encoding upward motion and you fire a few more action potentials, it makes it more likely that a subject's going to say motion is going upward than downward. If you fire a few fewer action potentials, it makes it more likely that the subject's going to say the opposite. Now, neither of those things is actually causal evidence, and the causal evidence comes from experiments in which people have inserted microelectrodes in the brain, listened in on neurons, and then activated neurons that all encode a particular direction of motion. So if you stimulate neurons that only encode upward motion, and you ask an animal subject to make decisions about whether motion is upward or downward, you can get them to say that motion is upward, even when the visual stimulus was moving downward. So together, these three different sets of methods, lesions, neurophysiological recordings that are correlated with behavior, and then manipulating activity to establish a causal relationship, is what has brought such power to our understanding of how the visual systems mediates actual perception. And it's these kinds of experimental maneuvers that I think are still waiting to be done for the mirror neuron system. Now, it's a particularly daunting task because the mirror neuron system is distributed through the brains in a broad swath. So doing the lesion or pharmacological inactivation experiments is virtually impossible. Why don't you agree with that?
Unfortunately.
Yeah. Even the single neuron recording experiments are challenging. You have to really think very carefully about what behavior you're going to measure that you're going to try to correlate neural activity to. It's a non-trivial experiment to design. And the same would apply to the manipulation of neural activity. So if you're manipulating one of these neurons that is firing when you observe an action as well as when you perform an action, the question is, what is the output of that neuron driving? what is the output of that neuron, what behavior is it driving, and designing an experiment that could test that explicitly is a non-trivial matter. I think these are why these experiments may not have been done yet, because they're very difficult to think about how to design. And they're probably easiest to design by sticking with the motor system that these findings were first described in. As you move to emotion, empathy, and aesthetic experience, I think the challenge gets even larger to how to design an experiment, especially in animal models, which is where these experiments probably would need to be conducted initially. But I think I'll leave my initial comments there. It's something for Vittorio to think about, certainly, and it hopefully provides a bit of hardcore neuroscience perspective on what we've heard about tonight. So,
because if we could spot the very same mirroring properties in a mice brain, that would probably enormously facilitate to implement many of the experiments you were suggesting, particularly for the neuropharmacological or genetic manipulation of this property. I mean, so one active goal is that to determine whether this property can be detected also in other mammals beyond primates. There is preliminary evidence, actually 2 papers appear, one in Science, one in Nature, showing that an audio-motor mirroring is present in singing birds. So if it is present in birds, I would be inclined to think that some sort of motor mirroring perhaps can be revealed also in rodents. And indeed, one of the first experiments we tried was to inactivate mirror neurons by means of injecting mascimol in the macaque monkey already many years ago in 1999. So what we were looking for was a double dissociation. I don't want to get too technical, but I mean, in the very same premotor area sit beside each other 2 different classes of neurons. The mirror neurons that you know, and another class of neuron that we call canonical neurons that have the very same goal-related motor properties of mirror neuron, but they are not excited by the observation of an action, but they activate when the monkey is merely looking at the object, provided that the object has the affordances compatible with the motor program controlled by the very same neuron. We injected mascimol in the bank of F5, where these neurons tend to be mostly segregated, and we had a visual motor deficit, and we published that in Brain in 2001. Fogassi was the main author of that paper. So after knocking out canonical neurons, the monkey is not paralyzed, it's not a motor deficit, it's a visual motor deficit, but shapes the hand in a very stereotypical way and finds its way to grasp the object only after exploring the object by means of haptic exploration. So the hand doesn't know beforehand, if you pass me the expression, how to shape to grasp the object, because the visual motor transformation is disrupted by knocking out temporarily this neuron. We injected the very same dose of mascimol in the convexity part of F5, where most mirror neurons tend to be clustered, and it was an action discrimination task. First monkey, worse than chance so we were incredibly excited. So we got the double dissociation, which is always what you're looking for in neuropsychology, but we couldn't replicate the results in the second animal. And the reason might be that although even the canonical neurons are a diffuse system, because they have a bilateral representation, there are canonical similar neurons also bilaterally in AIP. Why it works for canonical neurons and not? So one way to answer this question is because canonical neurons support visual motor transformation, but mirror neurons do not support action understanding. That would be the most negative conclusion one might draw from this failed unpublished experiment. On the other hand, I tend to think that that experiment was not well designed. So in other words, the monkey could differentiate different hand posture, also on a purely visual strategy, you see. So without implying an active motor resonance. There is indirect evidence that when you have a lesion affecting a part of the brain that normally show this mirror-like pattern of activation, you have problem in reconstructing the temporal sequencing of correlated action of humans. There is a paper published by Luciano Fadiga and colleagues, which appeared in Brain last year, on broadcast patients. These are aphasic patients that were screened, and none of which turned out to be positive for apraxia. So they had the typical standard of Broca aphasia, with reasonably spare comprehension but very poor language production. The test was very simple. They had a touch screen in which images were completely shuffled. By touching each of the image one by one, they had to rebuild the correct temporal sequence of two types of stimuli. Biological correlated action, a folk approaching a glass, grasping the glass, taking a bottle, pouring the water, bringing the glass to the mouth, or physical events like a bicycle standing straight and then progressively being more and more tilted and finally laying on the ground. Well, to make a long story short, Broca's patients have no problem in correctly organizing the temporal sequence of the non-biological action, but they were a chance in organizing the temporal sequence, the motor syntax of the correlated motor action. It's not what you're asking, definitely. But it's a close approximation, perhaps showing that this mechanism is relevant, not only for guiding your own action and production, but also to detect.
I think your instincts to try to get a rodent model working, because there you could possibly use genetic tools to specifically inactivate just the mirror neurons.
Yeah.
And that would be much more powerful.
Yeah.
But of course, the interest of mirror neurons for neuroaesthetics related above all to human responses. And so I'm just wondering whether we could get back. I mean, you just spoke about Broca's aphasic patients, but I wonder whether we can get back to the problem of mirror neurons in humans. I mean, it is difficult to imagine, you know, the experiments that turned out to be so influential about relating to a gesture or reaching for a cup. Rats that cannot engage in that kind of goal-directed action that is so important for the understanding of works of art. Yeah. So I wonder whether you want to move the discussion a bit to some of the difficulties with human.
Well, I think that Vittorio touched on it in his talk, in that the major method to investigate processing in human brains is fMRI, and it's been subject to a criticism that Vittorio mentioned, which is that the spatial resolution is such that you can have intermingled populations of cells that have very different properties, and you can't know that the same cell is doing these two different things, mirroring as well as action. And that's what was so beautiful about the neurophysiological recordings presented from, I guess it's probably epilepsy patients.
Yes.
And because they're able to, I haven't read that paper myself, but it sounds like they're able to replicate key components of the studies in the monkey.
Right.
And now the question would be that if you could really do that, then I think that is the best opportunity for rigorous mechanistic study in human subjects that would have to do with things related to aesthetics. Because I think one of the challenges of using that approach to study motor processes, motor mirroring, is that most of the places where you actually have epilepsy, the sources of epilepsy in the brain, are not probably where the mirror neurons are found in the ventral premotor cortex and the parietal lobe.
It's mainly in the temporal lobe.
That's right. But the epilepsy patients frequently have electrodes implanted in the medial temporal lobe, the structures of the medial temporal lobe. And it may be an opportunity there to especially look at emotion centers in the brain with respect to mirroring in ways that hasn't been done yet. I think that may be the most exciting opportunity out there. But these experiments are, I mean, there's only a few groups in the world doing experiments like this. You're working in a clinical population of ill patients, so you have, in some ways, less flexibility to study things rigorously in a behavioral fashion in these subjects.
but it's a huge opportunity in these handful of labs in the world that are doing these kinds of recordings. I don't know if you agree, but...
No, yeah, definitely. I think that these, as you said, provide the opportunity to study incredibly interesting brain regions like the amygdala and to a certain degree also the insula, while to have single neuron recordings from the ventral premotor cortex, I mean, it's probably one out of, I don't know how many, 100. It's very unlikely, unfortunately. Just before we move on to the discussion, in the rush to conclude, not to be too long, I made a terrible mistake, and I took full credit, which I didn't to have. I shouldn't have done it. The experiment which concluded my presentation was done primarily by Maria Alessandra Umiltà and Cristina Berchio in tight and constant collaboration with the Italian Academy in the person of David Freedberg, with whom we constantly discussed every single step that led us to finally approach experimentally this tricky topic. So sorry for not having mentioned, giving full credit to the people who really made the experiment and greatly contributed to make it possible.
Well, Vittorio, that characteristic courtesy is hardly necessary. I think all involved in the experiment learned a great deal, and now the rest of you know whom also to attack when you read the articles and have your doubts. Siamo qui presenti.